Now come to the another vector which is also one of the important vector known as expression vectors. Expression means after shuttle vectors the next vector which we have to study, these are expression vectors. Now what are these expression vectors? Let us see that what are these expression vectors. In addition to incorporation desired gene into the host cell, the objectives of recombinant DNA technology is to produce high amount of proteins encoded by the DNA insert. It means that our desire is to incorporate gene of interest in a host cell, but in addition we also want that the protein which was encoded by the DNA, ultimate product of DNA is a protein, that the protein which was encoded by the DNA of our interest, that DNA, it should produce high amount of protein, means the production of that protein should be in high amount. To achieve this goal, they introduce novel genes. Any particular novel gene must express. It means that the gene of interest, the gene which we are inserting in a vector, it must express itself in a vector. So how this can be achieved? Our next target is how we can achieve this target. So for this, expression of clone genes is carried out by inserting a promoter sequence. Insertion of promoter sequence. Now what is this promoter sequence? Promoter. Insertion of promoter sequence. Promoter sequence is basically the signal for initiation of transcription. What is transcription? Transcription means when our DNA converts into RNA, this messenger RNA, this process is known as transcription. So the sequence which is used in this process in transcription, we have to insert this sequence in our vectors. For example, this is a vector. Okay, if this is a vector, then we have to insert this particular sequence, which is insertion of promoter sequence, which is known as the promoter sequence in a vector, so that it can initiate transcription. And after that, one more sequence after promoter sequence, next should be the terminator sequence. Terminator means which can end this transcription method, means we have to insert two sequences, one sequence for initiation of transcription and another sequence for termination of this process of transcription, means this process should end. Now what is the use of inserting these sequencings? Because after transcription, when mRNA is formed, in the next step which is known as translation, this RNA converts into a protein. So we have to insert these sequences, means these are known as signals of protein synthesis. So the expression vectors are the vectors which contain signals for protein synthesis. Protein synthesis. And what are these signals? These signals are sequences, initiator sequence as well as terminator sequence for transcription, and after that sequence for translation. So expression vectors also contains marker genes such as ampicillin resistant gene is present in expression vectors, and other genes are also LAGZ gene, ORI region is present, somatostatin gene is present. No need to go in details about all these genes, but just what we have to write, if this question comes in our exam, just write about the expression vectors, that expression vectors are the vectors which contains signal for protein synthesis, so that we can get a particular protein, desired protein, in high amounts. And if you want to give example, वैसे example is, we don't have to learn this example in details, but still if we want to give, so we can give this example: plasmid, SOM 1. It contains promoter sequence for production of chain of somatostatin. Somatostatin is a gene known, so it contains the promoter sequence. This is one of the example which contains promoter sequence for a particular gene. So like this, expression vectors are the vectors. Just learn this definition, that expression vectors are the vectors which contain the signals for protein sequences. That means they have the promoter sequence for to produce a certain protein in large amount. These kind of vectors which have these signals are known as expression vectors.